Hi everyone, welcome to the channel. Be sure that you subscribe to our channel by just subscribing the bottom. You see there is a subscribe button before we started. Today I'm going to cover BSC 1010 General Biology 1, Chapter 15. It's chromosomal basis of inheritance. So this is about the Dr. Morgan's experiments at chromosomal theory at Columbia University. Dr. Morgan showed that Mendelian inheritance has its physical basis in behavior of chromosomes. Sex-linked genes exhibit unique patterns of inheritance, and then linked genes tend to be inherited together because they are located near each other on same chromosome. Okay, so chromosomes may be allosomes or sex chromosomes. In mammals, we have XX female and XY male. Okay, they can carry sex-linked traits besides determining the gender of the offspring. Chromosomes are a location of the genes, and they might be linked or unlinked. If they are unlinked, that's like typical Mendelian genetics. The independent assortment results in genetic recombination. Also, link can cause genetic recombination. They may cross over and results in genetic recombination. And then we can actually calculate recombination frequency, and then one percentage recombination frequency equals to 1 map unit, which is also equals 1 centimorgan. And we can actually map cytological maps and use it in the map location of the specific genes. Chromosomes also may have alterations in number and in their structure, such as deletion, duplications, translocation, and inversions. They can also have aneuploidy, such as trisomy or monosomy, or they might have polyploidy, very common in plants like strawberries, bread wheat. We see hexaploids in bread wheat. And also these trisomy and monosomy can cause genetic disorders. So let's take a look. Dr. Morgan used fruit flies as his model system, right? So why? Because they could be easily cheaply raised in the lab glassware. Female only mate once and lay hundreds of eggs, short generation time, and rapid experiments. And they had XY system, obviously. So a wild type is just a normal phenotype. Mutant is alternative wild type. So in this case, red eye color fruit fly is the wild type normal, and then white eye color is one of the mutants he created, and he crossed these two. When he crossed these two, F1 we see all red eye, F2 we see 3 red eye, 1 white eye fruit fly. So far looks like normal everything, but when he pay attention, he's seen actually all wide-eyed flies were males. That's because an allele for eye color on X, but not on Y chromosome. Okay, so today we can actually map different organisms, all the genes on chromosomes, and like you see here in this picture, human chromosome maps. Okay, so when he did his two gene problem with a homozygous recessive little v, little b, little vg phenotype crossing, so he was expecting 1:1:1 ratio, but actually he did not see this ratio because these two genes that he's studied were on same chromosome. So now we know that they are called linked genes, and recombination is basically that can be cause something not found in either parent, but we see it in the offspring. Okay, so the example here is a red and white parents, and they go through meiosis, and you can see here some look like parents, these two, and then other two don't look like parents. We call recombinant. So we can calculate this recombination frequency with the formula. Recombination frequency equals to total number of recombinants divided by total number of offsprings times 100. In this case, it's 17%, and we can actually use this to actually map the genes B, V, G, and CN, which is 17%. Student actually his student in Dr. Morgan's lab, he came up with the idea that frequency of recombinant offsprings, they are actually reflective distances between genes. So that actually opened up the whole idea of linkage maps. And now you can see an example of linkage map. Again, 1% recombination frequency now is called 1 map unit, is also a centimorgan. Okay, now we can actually make maps, design maps like this. You see here a fruit fly chromosome map with all these different genes in specific location. Okay, and there are 4 groups of organisms in this chapter that actually with the 4 different unique sex chromosomes. Human is XY system. Human female has 44 autosomes plus XX. Human male has 44 plus XY. Grasshoppers has XO system. Their male has 22 plus X. Their female has 22 plus XX. Some birds, fish, and insects, they have ZW system. Their male has 76 plus ZZ. Their female has 76 plus ZW. Bees and ants have haploid diploid system. Their male come from an unfertilized egg, which is 16 chromosome haploid, and their female come from fertilized eggs, which is 32 chromosomes and diploid. Very interesting. Okay, so X-linked genes, they are basically genes that they are located on X chromosome. Most known one is color blindness. Also 2 other diseases, hemophilia and Duchenne muscular dystrophy, are 2 other examples. X-linked dominant, there is very rare examples like Rett syndrome, which is a mental disorder. Alternation of chromosome number and structure. Basically, chromosome numbers can change. If we have an abnormal chromosome number, this is called aneuploidy. If you have a 2n plus, that's called trisomic. 2n minus 1 is called monosomic. So here is an example, Down syndrome on chromosome 21. We see an extra chromosome. This affects 1 out of 700 children born in the United States. Polyploidy is actually, again, common in plants, but we can see in one example here, a rodent. Here is the tail, very different than a regular tail. That's a polyploidy. Chromosome structural changes can be deletion, duplication, inversion, translocation. Deletion, if we delete one part, this is missing here. Duplication, if we duplicate it. Inversion, if we switch 2 parts. And then translocation, we have to have 2 sets of chromosomes and then translocate and exchange parts. And that's also, very lastly, there are genes that are actually called extranuclear, which is basically, they also non-Mendelian. They are found in mitochondria or chloroplast, and they don't follow Mendelian inheritance. So as you see here, spotted tomato leaves here, results from the genes in plastids. This is cytoplasmic inheritance, not regular Mendelian genetics. That ends our chapter 15. Happy studying.